Okay, good. Good morning, everybody. Good to see you again. In today's lecture, we should be talking about the transmission of information in the nervous system. So we're going to be talking about signaling in the nervous system. Obviously, you've heard quite a lot about quite a few signaling molecules, about some receptors. So today we're going to focus on how signal is transmitted in the nervous system, because there are differences and similarities with other tissues. Yeah, that's what we're going to be talking about. To start with, let's get a brief overview of when we talk about signaling. So what kind of signals we see in the nervous system, how they move, through which they move, through what they move, etc., etc. You've had lectures on the structure of some of the cells in the nervous system and of the nervous tissue. So what is the basic unit, or what are the basic units of the nervous tissue? Okay, we have neurons there, definitely. We have glial cells. We have synapses between neurons, okay? And there are some other things like blood vessels, etc., etc. Okay, the main cell types that we're going to be talking about are neurons, and we're going to talk a little bit about glial cells as well, because they do play a role in signal transmission and signal processing in the brain. So just a very quick review of what you know already. If this is a neuron, what are the bits of it? What is this? A dendrite, okay. What is this whole thing? Okay, the body of the neuron. But yeah, everything was correct. What is this? That's the nucleus. Do we have anything else in the cell body? I just can't hear you. Like you have to shout. ER. Okay, definitely. I'm not going to draw that, but yeah, there's some ER in there.
It's the only place where the Golgi is, right?
There's definitely some Golgi apparatus. Yeah. There's some mitochondria there, of course. All sorts of things. Yeah, definitely. There's a cytoskeleton. Good. Okay. Lots of stuff in the cell body. What is this? That's the axon. Okay. So that's kind of the general shape of a neuron. Obviously, and you probably know that you've seen different kinds of neurons have different kind of structures. The dendrites can be branched. The axon usually isn't. Of course, because we're talking about the transmission of information, the neuron, as I've drawn it, doesn't just sit there on its own, but obviously it's connected with other neurons, and it's surrounded by glial cells. So the connections, as you well know, are between axons and dendrites. So basically, if we continue this way, we'd end up with a synapse coming from other neurons like that. And the reason why I'm drawing that is that it's quite important to keep in mind that it practically never works that you have one neuron communicating with just one neuron. Okay? So I drew quite specifically quite a few different axons that connect to the same neuron. And so it's important to keep in mind that neurons are not just simple on-off switches, but they actually integrate several different signals in order to produce a signal that goes into the axon. Okay? I'm not going to spend a lot of time talking about that, but do you understand what I'm saying? There are several different information inputs into each neuron, and quite a complicated decision-making, in a way, is taking place in the dendrites and the body before an action potential, which we're going to talk about in a minute, is sent through the axon and is sent to a different neuron. Okay? So neurons are not just simple on-off switch. There's a lot of integration, quite a complicated decision-making process, whether to send a signal or not. Right, I've been talking about signals. What modalities of signals can we find in the nervous tissue? Somebody, yeah? Neurotransmitters, okay. Okay, there's excitatory, inhibitory, there's something neurotransmitters, neuromodulators. Okay. This all is one big group of signals. What's...
Chemical and electrical?
Yeah, that's exactly what I was looking for. And yeah, it's always funny to ask a very vague question so that people come up with some interesting ways of understanding it. But absolutely, that's what I was looking for. So we have electrical signals and we have chemical signals. Now, today we're going to talk about both of them. We're going to start with the electrical signals, and then we're going to go on to the chemical signals and some of the molecular biology or molecular biochemistry of how they work. In order to create and transmit electrical signals, what do we need for that? We need ions, absolutely. Okay, if we take a solution of salt, there are plenty of ions, but we can't really create, we need a concentration gradient, okay? And a concentration gradient, okay, sorry, you have to, yeah, one at a time.
A membrane which separates.
Okay, we have a membrane which separates two compartments with creating a concentration gradient? We need a potential difference, okay? So all the things that you mentioned are obviously correct, but all these are necessary to get a potential difference because that's what enables us to create electric signals, electromagnetic signals. So we need a potential difference. The potential difference on a cellular membrane is usually just called a membrane potential, even though it's actually a potential difference, but we should just call it a membrane potential. And we talked quite a bit about membrane potential in mitochondria, and you've heard about membrane potentials in other tissues as well. So let's very quickly talk about how potential difference on a membrane actually arises in our cells. How do we get a potential difference to start with? Okay, we have active transport of ions. Can you elaborate on that? How that creates a potential difference?
There's an electrochemical gradient of sodium outside there's a high level of sodium on the outside and a low level on the inside of the cell.
Okay.
And
that's true.
Slightly lower concentration of potassium on the inside, so the inside is more negatively charged than the outside.
Okay, so your suggestion for the cause of existence of a membrane potential is that there is more sodium outside, less sodium inside, and there is slightly, what was it with potassium?
There's obviously more potassium in the inside relative to the outside, but in total charge level there's less potassium. There's less positive charge on the inside than there's on the outside.
Okay. So let's take this as a cell, as our neuron, it doesn't really matter. You said correctly that we have quite a lot of sodium outside, not very much sodium inside. We have quite a bit of potassium inside, and not that much potassium outside. Any other differences in calcium? How is it with calcium?
Outside. More than...
Okay. Here, the difference is actually by far the biggest one of all the ions. Okay. Well, the differences here are about 100-fold 50-fold or something like that. Here the difference is about 4 orders of magnitude. Okay, so the difference in calcium concentration outside and inside is absolutely huge. But you've heard about that calcium as an important second messenger. We're going to talk about it today a little bit more as well. Any other ions that may play a role? Okay, how's it with chlorides?
Concentration is high outside.
Okay, so we have quite a lot of chloride outside, not a huge amount inside. Okay, those are the ions that I think that's enough. Obviously, there are differences in magnesium, etc. But those are the ions we're going to be interested in, let's say. Right, so we have these differences to start with, let's say. We have these differences in concentrations of ions. But this doesn't actually tell us anything about a potential difference. I mean, you correctly said that there are actually different numbers of positive and negative ions outside and inside. But let's start with just purely concentration differences like this, okay? There is no potential difference between outside and inside at this point, okay? We only have differences in concentration, but overall the sum of negatively charged and positively charged ions is the same outside and inside, okay? Now, how do we create, how do we give rise to a membrane potential in a situation like this?
Pump the ions against their concentration gradient.
We're gonna pump ions against the concentration gradient, okay?
Yeah, for example, Na plus to the outside
Okay,
and K plus to the inside.
okay, right. So we take one sodium, take it outside, one potassium, put it inside. How does that create a membrane potential? Okay, right. I understand that you mostly are trying to get into NAK ATPase, which is indeed electrogenic because it transports more sodium out than it transports potassium in. You've heard of NAK ATPase before? Yeah? Okay. That's a good point, and that is part of the mechanism how we create membrane potential in excitable tissues. But actually, there is a simpler and probably much more important mechanism how to get the membrane potential without any use of ATP. Well, not any immediate use of ATP. Okay. In a normal resting cell, for example, a resting neuron, we can imagine that the cell membrane is only permeable to potassium. Okay? It's not completely true, but it's mostly true. Okay? So the only ions that can move from the outside or from the outside in or from the inside out are potassium ions. Okay? So only potassium ion channels are open. What happens in that situation? Okay? We have a fair bit of potassium in, not that much potassium out, and we open potassium ion channels in the membrane. What happens? Potassium goes out. Okay? Why does potassium go out? Okay, it's along the concentration gradient. Can anybody explain why? Because the individual potassium ions have no idea about any concentration gradients. Okay? They don't understand this concept. Okay? So how do they know that they should be going out of the cell and not coming into the cell?
So just a case of probability.
Okay, can you explain that?
Well, it's more probable that potassium will
More collisions inside the cell, more collision of potassium ions, so more of them actually get pushed out of the cell rather than on the outside where they don't interact.
There are more collisions of potassium ions in the cell, so that more of them are pushed out. Okay, the total number of ions, we agreed, is the same inside and outside. So it's not so much, I mean, you're both right, but let's try to explain for everybody so they understand why that happens.
the probability of potassium leaving the cell is bigger than the probability of potassium entering the cell.
And why?
Because the concentration is larger.
Okay, we're starting to move in circles. But I mean, yeah, you are all correct. Okay, so let's imagine we have, this is the membrane, and we have 3 potassium ions here and 50 potassium ions there. Okay, now they're moving just randomly around. Okay, they know nothing about anything. They're just randomly moving around. Okay, some of these random movements will find a channel and will allow them to go to the other side of the membrane. Okay? Now, the probability of each individual ion to cross to the other side is the same on both sides. Does that make sense? Yeah? Because they're just moving randomly. So there's no difference. So it's not so much about that there are just too many and they try to push out. Okay? Yeah, I'm not sure if that's what you meant, but that's how I understood it. They're just randomly moving, and the probability of moving to the other side of each individual ion is the same. But there are many, many more ions here. So the overall probability of one potassium ion moving this way is much bigger than the probability of one potassium ion moving that way. Okay? Just raise your hand if you understand this. Okay, that's reasonably good. Okay, so it's absolutely correct to say it's because of the concentration gradient or because it's because of entropy, because obviously having more ions here and fewer ions there is a more ordered system. So entropically, they will try to move around. But I think the best explanation is by means of statistics and just random movement, and I think it kind of makes sense why they're going to move out. Right, so potassium ions are moving out of the cell. What happens with membrane potential? Potential difference on the membrane.
Decreases.
Okay, it becomes more negative inside. Okay, decreases, well, it depends how you measure it, but it becomes more negative inside because we're losing positive ions from the cell. It becomes more negative inside. Okay, and this is the main mechanism how we get a resting membrane potential. Okay, right? So at rest, the membrane is permeable only to potassium ions. They leak out, and this means that the inside of the cell becomes more negative. Right. How much more negative is the inside in an average neuron? Minus 75. Okay, let's agree for biochemistry. Let's agree on minus 60 to minus 70. Okay? Those are reasonably good numbers for a neuron. There are different cell types, like skeletal muscle has actually much more negative potential. It goes down to minus 90 or minus 95. But let's, you know, as a kind of consensus value, let's have, okay, minus 60 to minus 70 millivolts, negative inside, okay? So that's the resting potential. The neuron is just sitting there, and it has this kind of potential on the membrane. That on its own is not moving any information, okay? That's just a stationary cell that doesn't do anything. So what we need is, we need to create some disturbance in this resting membrane potential in order to transmit this disturbance as a signal, okay? As a 1 and 0, like if we look at it from a digital point of view, okay? So resting potential would be a 0, and basically moving something, some other kind of potential would be a 1. So what is this other kind of potential that that I want to be talking about? The action potential, okay? So this is resting, and now we're interested in the action potential. What is an action potential? What is an action potential? Well, let's continue talking about neurons. What is an action potential? It is?
Influx of positive ions.
Efflux or influx? Influx of positive ions. That's true. Yes, that's correct. In terms of membrane potential? It's a depolarization. Okay, so correct. The influx of positive ions causes depolarization, and it's the membrane depolarization moving along the axon or along the dendrites that is the action potential. That's the actual signal. Okay? So action potential, and you correctly said that it's caused by a movement of positively charged ions into the cell and depolarization. Right, how does that work? First of all, what kind of positive ions flow in? Okay, it's mostly sodium, but we can hear that it's calcium as well, depending on what causes the action potential. And how does it work? How does it come about that there is an action potential suddenly? Okay, ion channels are open, and we just mentioned that they're going to be mostly sodium ion channels because it's mostly sodium going in. Why do these channels open? They're voltage gated. Okay, so why do they open?
After it goes under the threshold, they just open.
Okay, goes above the threshold, but that doesn't really matter. Okay, so we have voltage-gated sodium channels. Something happens to the voltage. How come there is a change in voltage anywhere or membrane potential? Yeah.
Calcium channels are stimulated by neurotransmitters, or neurotransmitters go to receptors and they stimulate opening calcium channels.
Okay, so you said correctly that most of the action potential is done by voltage-gated sodium channels, and we're going to talk about them. But the first stimulus for this depolarization that you talk about, this moving above the threshold, in order to open voltage-gated sodium channels, there needs to be some stimulus. And the stimulus usually comes from ligand-gated ion channels, either sodium or calcium, that cause a little bit of depolarization, which is enough to start the action potential going through voltage-gated sodium channels. I'll explain it in more detail straight away, okay? So if you don't understand what I'm talking about, I'm going to draw a schematic to show it. Well, actually, I could have used this, maybe. So what happens here is some kind of depolarization comes here, something happens on the synapse, and we're going to be talking about it in the second half of the lecture, what happens here. But what we get is that some neurotransmitter activates some kind of ion channels, ligand-gated ion channels. You know the term, ligand-gated ion channels. Yeah, okay. Which cause some kind of depolarization, so the potential here rises a little bit from -60, -70. And that opens voltage-gated ion channels, which then propagate the signal further. Okay? Does that make sense? Okay, it's just so trivial that you're kind of bored. Yeah? Okay, good. So let's now talk about these voltage-gated sodium channels, because they're actually quite interesting and they're very important for signal transmission in the nervous system. Yeah?
Ligand-gated, is it for potassium or sodium?
There are all sorts. Mostly it's for sodium or calcium, but we're going to be talking about the specific ligand-gated ion channels in future lectures, okay, in a couple of lectures. We're going to be talking about all the different kinds of ligand-gated ion channels. So today's lecture is not going to be very much about ligand-gated ion channels. I just mentioned them because that's the first stimulus for the action potential to get going, okay? But my focus now is on voltage-gated, and specifically voltage-gated sodium channels. Okay, this is a voltage-gated sodium channel. This, what do you think is this? That's the membrane, okay, and this is the actual channel with a pore through which sodium can flow in. Okay, so this is outside, this is inside. Now, it's a voltage-gated sodium channel. Okay, it's gated. We need to have some kind of a means of opening and closing it. And since it's a voltage-gated ion channel, we need this gating mechanism to be sensitive to voltage, to the potential difference on the membrane. So let's take 3 minutes and try to talk to people around you and try to come up with a mechanism how we could, you know, if you were to design a voltage-gated ion channel, how would you do it? how would you make a protein that responds to changes in voltage by opening and closing a pore through which ions can go? Okay, so let's 2 or 3 minutes, just talk and try to come up with a mechanism. I think the time's up. So whoever wants to start, just give me some ideas. How would you design a voltage-gated ion channel? You want a protein that opens when the voltage changes.
You need something that attracts positive ions, so something negative maybe?
Something negative? Okay, like what negative? Amino acids? Okay. Okay, so what kind of amino acids would they be? Okay.
Cytosine, hydroxy.
Okay. Just any amino acid that might be negatively charged. Aspartate, okay. Glutamate. Okay, at least those. Okay, so we have negatively charged amino acids. Okay, so we have protein with some negatively charged amino acids. What then? What would you do with that? What would you do with that? You have negatively charged.
Oh, sorry.
You have negatively charged amino acids. Okay.
Then all of a sudden you need something positive to repel the thing negatives.
Okay, so you want both negatively charged amino acids and something positive?
Top, and as it goes through the tunnel, it gets more and more positively charged. So,
Yeah?
I think the protein has to have some charge itself for it to be able to respond to charge from the outside.
Okay, yeah. So we need at least part of the protein to be charged, negatively, positively, or both. Okay. Any other ideas? Did anyone come up with something completely different? No? Okay, that's pretty much how it works, not surprisingly. It's a bit simpler than what you mentioned. You don't need that many different charges. How it actually works is that there is a domain which is part of the ion channel that is positively charged. Okay? How do we make, so we talked about negatively charged amino acids. Do we have any positively charged amino acids? We do, excellent. Which ones? Indeed, yeah. Which are? Lysine, arginine, maybe some other ones. But in this case, it's actually arginine residues. There are several arginine residues that are positively charged on the domain. And as you correctly described the mechanism, under normal resting potential, there is relatively large positive charge outside, which is pushing the domain down. Okay? You may recall from one of my lectures about mitochondria that the membrane potential in mitochondria, which is about, let's say, minus 160, so quite a bit bigger than this one. But still, the membrane potential, when you actually expand it to macroscopic dimensions, is absolutely massive. Okay, we're talking about millions of volts per meter or something like that. Okay, huge electric fields. So quite literally, the electric field at the resting potential level is pushing down this protein and is keeping the pore shut by this other part of the protein. Okay, now this is actually a schematic. It doesn't look exactly like that. Okay, but it works. The mechanism is exactly like that. So what happens when we depolarize the membrane? So suddenly, there's much less positive charge here, or the difference in the charges is smaller. So what happens is that this positively charged domain is now free to move up, because it's no longer pushed down. And the channel's open. Okay? So a relatively simple mechanism. Right, what happens next? We have sodium channels open. Sodium is flooding into the cell. The membrane is depolarized. So the membrane potential is getting higher and higher. What's next? Indeed, yeah, the signal, the depolarization propagates. And I'll just mention one thing. Why does it propagate just one way? But we'll answer it in a minute. That's fine, that's all happening. But what we're leaving at this current state, what we're leaving behind is massively depolarized axon with sodium channels open. Yeah?
Because some areas need some time to repolarize.
Definitely. We probably should repolarize the neuron in the end so that we can use it again.
Yeah, so the areas that are left are...
Oh, you're answering the question why it only goes one way.
Yeah.
Let's leave that aside now, okay? Because how do we close the channels? How do we close these channels?
It's another threshold, isn't it?
It's another threshold.
Another threshold. And then the pump
What leaks back?
Because now you've created another gradient in the opposite direction.
No, not really. We haven't really created another gradient because what we're doing, we're dissipating the gradient of sodium. So the gradient of sodium is getting smaller and smaller and smaller. Okay?
Yeah.
That's what's happening. But it's not reverse. It will never reverse just by letting sodium in. Yeah? So how do we repolarize it? How do we close the channels? Okay, so there are some potassium channels, and it's actually quite complicated because in neurons it doesn't play a huge role, but okay. The trick here is that these voltage-gated sodium channels will just close on their own after some time. And I'll just explain why it's so important. So, in fact, there's another gate in the channel, which after about 1 or 2 milliseconds, automatically closes the channel. Okay, so there is no need for any other changes of voltage or potential or anything like that. Okay, after the channel opens, it's open for about 1 or 2 milliseconds, and then it automatically closes. Okay, so we moved from here to here. And now we've closed the channel. Okay? Do you understand this mechanism? Okay. It's called inactivation, and it's actually the essential thing for ensuring a one-way propagation of the signal. Because once these channels are inactivated like that, you can't open them by depolarizing the membrane. Okay? Because it's a completely different, it's an independent mechanism. So first we open them by moving this positively charged domain. Okay? We open the channel because of which is caused by depolarization. But the channel itself automatically closes after 1 or 2 milliseconds. And something else has to happen for it to be able to open again. Okay? The something else is actually repolarization. So I'll just kind of put it in one maybe long sentence. The voltage-gated sodium channels open on depolarization. Okay? There's a positively charged domain which moves out, opens the channel. Sodium ions can flow in, but only for 1 or 2 milliseconds. After that, the channels close, which allows the cell to repolarize by means of opening potassium channels, by means of some sodium, some ion pumps, etc., etc. And only after repolarization do the ion channels, the voltage-gated ion channels, get to the original state. Okay? So when you started talking about why it moves only one way, you said correctly that basically there is an area, so if this is the action potential, there is an area which is normal polarity, normal membrane potential. Here is an area of a cell that's depolarized. But obviously here there's going to be another area of the membrane that's already repolarizing. Okay? So you could still, what could happen still is that the signal will start moving that way because there's depolarization here and there's repolarized, well, almost repolarized membrane here. Okay? So there is a time, there's basically a time lag that's built into the system. So it doesn't allow situations like that where you have depolarized membrane and repolarized membrane next to each other where they can just go. Okay? So 1 or 2 milliseconds is enough to separate these two areas so that the signal moves only one way. Okay? Don't have the feeling that it makes sense to most of you. Not really. Okay. Let's now leave aside the idea of unidirectional movement, okay? Because that's not that important. Does this cycle, the cycling of voltage-gated sodium channels, does that make sense? Okay? So that's the important thing for now. So there are 3 different states. Normal repolarized ion channel that's closed, then it opens, then it's inactivated. Okay? 3 different states of voltage-gated sodium channels. And why are they important? Well, they're important because they transmit the signal, so we need them for action potential. But the other reason why they're important, why we talk about them here, is that they're also pharmacologically important. And we're going to talk about the pharmacology of voltage-gated sodium channels in a later lecture, but I'll just mention that local anesthetics bind and block voltage-gated sodium channels. Okay, local anesthetics, does everyone know what they do? Yeah? No? Okay, okay. Local anesthetics are basically drugs that usually get injected where you get when you're getting a small operation, a small surgery, or when they're extracting a tooth. Okay, that's what they inject. So it's a local anesthetic. It only blocks pain in a limited area of your body and doesn't cause you don't lose consciousness. Okay, so there are general anesthetics where you sleep through the surgery. The local anesthetics where you just don't feel part of the body. Okay. So local anesthetics work by binding and blocking these voltage-gated sodium channels. But we'll talk about that later. The other interesting compound, but that's not used pharmacologically, that also blocks voltage-gated sodium channels is something called tetrodotoxin. Has anyone heard of tetrodotoxin before? Never heard? Okay. Have you heard of a fish called fugu? Never heard of it. Fugu? No? Okay. Fugu is a delicacy in Japan, and I think nowadays it gets exported elsewhere. But the trouble with fugu is that big parts of the body of the fish contain tetrodotoxin, which is many times more toxic than cyanide. So when you eat fugu, you have to be very, very, very careful to separate the meat that doesn't contain the toxin from all the entrails that contain the toxin. And there are there are several cases a year, and many have been reported, where people just die after eating the fish. So I'm quite surprised that no one's ever heard of it. Okay, some people have heard of it. Okay, it's something that often sometimes you find in newspapers where they say, oh, people in Japan, oh, they're so stupid because they ate this. I mean, actually, apparently, if you go to a good restaurant where they can prepare the fish properly, there's actually very little risk of dying. But anyway, the other interesting thing is that it's actually not the fish that makes the toxin. There are actually bacteria in the fish that make the actual toxin. And the fish just basically, it's a symbiotic relationship of the fish and the bacteria. So the bacteria protect the fish from predators, because obviously predators, if they eat the fish, they die. So there are actually, apart from fugu, the fish, there are actually several other organisms. There are other fishes and some amphibians, I think, that contain tetrodotoxin as well. So, okay, it's produced by bacteria. But anyway, the reason why tetrodotoxin is so deadly is that it binds to voltage-gated sodium channels and blocks them. And since we just said that voltage-gated sodium channels and their opening is essential for basically all signal trafficking in the nervous system, obviously you're in trouble when that happens to you. What do you think is the cause of death in tetrodotoxin poisoning?
Suffocation.
Yes, you suffocate. Because the parts of the nervous system that are easiest for tetrodotoxin to get into and to block is the peripheral nervous system. So basically, the first symptom, and the thing that kills you, is paralysis. It's complete paralysis. Okay, because tetrodotoxin blocks all the signals in the nerves. Okay, so you die of suffocation because you can't breathe. Now, when I had a lecture for Czech students, they were asking, well, because what you usually say and what you read is that generally people who are poisoned by fugu, they're actually conscious most of the way, and actually it looks like they're conscious pretty much before they die, which obviously is quite unpleasant, I imagine. But then the Czech student said, well, you know, if tetrodotoxin blocks all signals in nervous tissue, how can they be conscious? Surely their brain is blocked as well. Here the difference is just how quickly the toxin can access different parts of the body. And since you've heard about the hematoencephalic barrier in the brain, yes, okay, the transport of the toxin into the brain is slower than to the peripheral nervous system. So the paralysis comes much faster than the loss of consciousness. Okay, so tetrodotoxin and fugu fish has some interesting connection with voltage-gated sodium channels. Obviously, tetrodotoxin is not used pharmacologically, but it has been used and still used in research, because having a very specific blocker of an ion channel is a very important research tool for manipulating or finding out various things about nerves. Last thing I'll say, just for your interest, the lethal dose of tetrodotoxin is 25 milligrams when taken orally, and 0.5 milligrams when injected. So it's a fairly potent poison. Right, questions before a break? Okay, can you shut the door? All right, before we continue with the second half, of the lecture, let me remind you once again, if I say, do you have any questions? If you do have any questions, ask then, and not during the break, because lots of people will have exactly the same question as you have, but since I only replied to you, they won't know that, okay? So when I say, do you have any question? If you do have any question, ask then, because then I can explain it to everybody. The things that seem to be a bit confusing, or some people seem to be, unclear about it. What I described here with the voltage-gated sodium channels, that's not the beginning of the depolarization. In order for this to happen, we already need the membrane to be at least a little bit depolarized. Okay? As James mentioned, the threshold. The membrane potential needs to move to more positive values until it reaches a threshold when the voltage-gated sodium, okay? So something else first needs to depolarize the membrane, at least a little bit. Not completely, but at least a little bit, okay? And we talked before that there are some ligand-gated ion channels or some other ways to start the depolarization. So what I described here, and what the role of voltage-gated sodium channels is to propagate the action potential, not to create it, okay? So first there needs to be some stimulus to start this whole process. Okay, so that was one thing. Now the other thing was how does it work with the unidirectional propagation? I tried to think about how to explain it cogently. So if this is the axon, the action potential is going to look something like this. This is the action potential. This is repolarized membrane, and this is also repolarized membrane. This is depolarized membrane. Okay, I'm using the same convention as I did before. So there are lots of positive charges in the normal resting state. Okay, because we have -60 negative inside, so it's quite a lot of positive charge. During depolarization, let's say there is no charge. Okay. It actually may go to the other end. It doesn't matter. Now let's not talk about the specific numbers. But let's say that there is no potential, okay, during depolarization. No potential difference. Do you understand what I'm talking about? Okay? So let's imagine that this is the movement of, or rather this is the snapshot of the action potential on an axon. So we have normally repolarized resting state membrane. We have repolarized membrane after an action potential. And we have the bit that's actually moving, that's the signal. This is the action potential. That's the little bit of the membrane that is depolarized that's moving along the axon. Okay? Now, if there was no inactivation, no automatic inactivation of the channels, there would be absolutely nothing to stop the action potential moving the other way. Because this membrane is already repolarized. Does it make sense? Yeah? So what we need is the automatic inactivation. So in fact, this is resting. This may have resting potential, but it's actually inactivated. Well, it's not probably quite resting potential. It's a bit lower than that, okay? Because if it were resting potential, it wouldn't be inactivated anymore, but that's fine. And now the rest of it is resting again, not inactivated.
How long is that? How long are they inactivated?
They are inactivated until they repolarize, until they completely repolarize, okay? But if they didn't inactivate, we wouldn't be able to repolarize the cell, because there will be sodium flowing in all the time, okay? So we need to close them by a different mechanism than voltage. If we just relied on repolarization, it would never happen, okay? Okay, so there are two reasons for closing them automatically. First of all, in order to repolarize, because otherwise we would just expend huge amounts of energy to overcome this amount of sodium ions flowing in. I don't think that would be even possible. And the other reason is that it allows us to that it allows the action potential to move one way. Okay, is it a bit clearer now?
And by inactivation you mean closing?
That's this, yeah. So the channel closes, but by a different part of the protein, by a different mechanism than it was closed originally. So it starts being closed by this voltage-dependent domain, but in the inactivated state, it's closed by a different part of the protein. And it needs to repolarize in order to get to the original state so that, again, it can open. In the inactivated state, it can't open. Okay? There's no way to open it. Okay? Yep.
Yeah, there are some viruses that make the signal go back to the nucleus of the neuron. So how is that possible?
There are some
Viruses.
viruses
Yeah.
that make the signal go the other way around.
Yeah. I think
Okay, I've never heard of that. I know of viruses that move from the synapse into the body. The virus itself, or the virus DNA or whatever, can move into, yeah, that's quite a common thing for some neurotropic viruses. But that has nothing to do with moving the signal. Now, of course, and I don't really want to complicate the picture even more, but if you start an action potential here by some method, like having a synapse here, which actually isn't very common, or by putting some electric discharge there, the action potential will obviously move in both directions. Okay? But that doesn't happen often. Okay?
But when it's inactivated, then it also has to switch back to the original.
That's right. And that happens on repolarization. So the channels are closed, potassium channels open, ion pumps start pumping, and once the potential gets back to, let's say, -60, this configuration changes into this configuration. The channel is still closed, but it's ready to be open. While in this configuration, you can't open it. Okay? Right. Okay. So this was voltage-gated sodium channels, and this whole thing basically describes the movement of the action potential through the axon. But obviously, we said in the beginning, neurons communicate between each other. So what we're going to be talking about now is the synapse. What happens at the synapse, and how signal is transmitted between two different neurons. The first thing I want to mention is that there are two kinds of synapses, two general kinds of synapses in the, well, let's say human nervous system. Does anyone know what two different kinds of synapses we have? Okay, so interneuronal and then between neurons and other tissues, for example, the neuromuscular junction. Okay, that's one way of dividing it. Depending on the type of signal. Okay, True. Excitatory is, yeah, okay. Inhibitory. Good. But remember we talked about two different kinds of signal in the nervous system in the beginning? Okay. They're chemical and electrical synapses. Okay? We're going to be mostly talking about chemical synapses, but let me just mention that there is such a thing called electrical synapse. It exists even in our nervous system, for example, in the retina. Some of the cells in the retina are connected via electrical synapses. How would an electrical synapse work? How would you connect two cells so that they transmit electrical signal to each other? Excellent. Okay? It's via gap junctions. So electrical synapse is nothing else than two cells connected by gap junctions. Obviously, gap junctions, as you know, allow ion currents to flow through. So any depolarization, any electrical signal will just flow through into the other cell. Okay? Outside the nervous system, you find gap junctions and, in effect, electrical synapses, for example, in the heart. Okay? Okay, myocardial cells are connected by gap junctions and they transmit the signal for the contraction by this means. But that's all I'm going to say about electrical synapses because in the nervous system they're actually very rare. So let's now talk about proper chemical synapse. Let's very briefly review the structure of the synapse. So what is this? Okay, presynaptic neuron. This obviously is going to be the postsynaptic neuron. Now, what are the interesting structures we find in the presynaptic neuron? In the terminal button. Okay, definitely. Now, this drawing is not to scale, okay? Very good.
Mitochondria.
Thank you. My favorite organelle. Okay, some cytoskeleton and all sorts of other things. Okay, this is pretty much what we're going to need now. So talking about the presynaptic, the terminal button. What are the mitochondria there for? Why do we need mitochondria in the synapse? For the transport. How do mitochondria transport?
The energy for the transport.
Okay, so we need mitochondria in order to provide energy for all the processes that are going on there, definitely.
And ATP for channels.
Absolutely, ATP for all sorts of things. Any other role for mitochondria in the synapse? Mitochondria play quite an important role in the metabolism of some of the neurotransmitters. Okay? So I'm not going to go into details. You've heard some and you will hear some more about that. But mitochondria degrade and also help synthesize some of the neurotransmitters. Okay? So they're needed for that. What about the postsynaptic neuron? Yeah? Yeah?
Are they not made, though, in the...
Yes. Yeah. Mitochondria, well,
So
I
they
can't...
play that specific role in this postsynaptic area, per se?
Okay, say again. Say again.
Would that specific mitochondria there play that role of making and making neurotransmitters?
Yes, because the pathways, let's say mitochondrial pathways that play a role in the metabolism of neurotransmitters are general. They just happen everywhere. So if all other stuff, the raw materials and stuff like that are in place, mitochondria will just do it. Okay, so you don't need any special kind of mitochondria. Okay. Yeah.
What's the point of the transport then from the body to the axon terminal?
The transport of what?
Of the neurotransmitters?
Oh, I thought you were talking the transport of mitochondria.
No.
Most neurotransmitters produced in situ. It's not produced. Proteins are produced in the body, but non-protein molecules are produced mostly in the synapse. Okay. What do we find in the postsynaptic neuron? Okay, definitely receptors.
With some channels that are sensitive to what's attached to the receptors.
Okay, and we call them ligand gated. Okay, so some of these receptors presumably are going to be ligand gated ion channels. Okay. So yeah, we have receptors, and some of them are going to be ligand gated ion channels. Some of them will have some other signaling pathways. Okay, anything else we need there?
And there are also degrading enzymes produced that degrade like chemicals which are not used.
Yeah, very good. So we have enzymes that degrade neurotransmitters, and the one we've talked before about is acetylcholinesterase, and that's actually present on the postsynaptic membrane. Yeah?
There's also a pump that comes back to the
Very good. So one of the things we didn't talk about is the reuptake of neurotransmitters back into the presynaptic neuron. Okay? Okay, so we have transporters for either complete neurotransmitters or bits of neurotransmitters back into the presynaptic neuron. And while we're talking about the presynaptic neuron, there are actually neurotransmitter receptors, so-called presynaptic receptors, on the presynaptic neuron. What do you think is the role of receptors on the presynaptic neuron? Not really for the reuptake. There are transporters for the reuptake. Okay, so there are feedback loops. There's feedback going back into the presynaptic neuron, basically telling the presynaptic neuron whether it released enough neurotransmitter, too much neurotransmitter, or too little neurotransmitter. Okay, so presynaptic neurons presynaptic receptors are very important in basically gauging the amount of neurotransmitter that's been released. And in future lectures, we're going to talk about some of them being very important in pharmacology, because by influencing the presynaptic receptors, we can actually influence the amount of neurotransmitter that gets released. Okay? So presynaptic receptors, feedback receptors for the amount of neurotransmitter. Okay, I'm fairly happy with what we've drawn so far. There are probably going to be some ion channels here as well, and probably there are going to be some voltage-gated sodium channels there as well, but that's not that important. There is one structure or several structures that are completely missing, and that are, as I mentioned in the beginning of the lecture, are quite important for signal transmission in the nervous system, and that's glial cells. Okay? What kind of glia do we have in the brain, in the central nervous system? We have oligodendrocytes, astrocytes, microglial cells. Okay, let's forget about microglia. They're not very interesting. Let's forget about oligodendrocytes, because the role for oligodendrocytes is just to make myelin sheaths. I mean, it's a very important thing, but not here. But astrocytes actually have projections that are in close contact with synapses. And the role, in the past 10, 15 years, the role of astrocytes is, it's becoming clearer and clearer that astrocytes actually are very important for signal transduction, and not only that they actually, not only that they influence the signal transduction in the synapse, but they can actually transmit signals on their own. Okay? Do you know, I don't know if, well, let's try it. Do you know what is the ratio between glial cells, the amount of glial cells and neurons in our brain? It's about 10 to 1, okay? There are 10 times as many glial cells as there are neurons, okay? So even though all the attention goes to neurons, and of course they are very important, there are actually 10 times as many other cells in the brain, which we know relatively little about. But it's getting clearer and clearer that astrocytes not only take up neurotransmitters from the synapse and then recycle them or degrade them depending on the specific neurotransmitter, but they also have receptors on their membrane. So they can actually detect the release of a neurotransmitter and react to it. Now, astrocytes cannot produce an action potential. Okay? So they don't do all the stuff that we talked about in the first half of the lecture. But what they can do upon binding a neurotransmitter, they open calcium channels and allow calcium to flow in. And this calcium then, these calcium waves move through the cell. Okay, again, this is not to scale, okay? The synapse is actually tiny compared to the astrocyte. Okay? The astrocyte is a properly big cell, okay? So if it were to scale, the synapse would be something like this, okay? Yep, just so that you know. Okay, so the calcium wave that's created by the astrocytic receptor binding a neurotransmitter moves through actually a pretty large area of the brain and can influence other synapses that are elsewhere. Okay, metabolically or by secreting some other signaling molecules. Okay? I don't want to go into huge detail. What I want you to remember is that astrocytes are very important. They communicate with synapses, they communicate with neurons, and they probably are important in what goes on in our heads. Okay, the last thing I want to talk about is how we actually, so we talked about the action potential. So the action potential is moving down the axon. And now I want to talk about how the action potential is translated into neurotransmitter release. Okay, because that's the next step in signal transmission. Okay, we need to release neurotransmitter so that the signal can continue to the next neuron. So how does it work? The process by which neurotransmitter is released from the vesicles is called exocytosis. So let's talk about exocytosis in the synapse. Okay, what do you think what this is? That's the vesicle, and that's so that's the cell membrane, that's the vesicle. Again, not to scale, obviously. So we have a neurotransmitter here. Now, the actual signal for exocytosis in the synapse, and actually for exocytosis in most tissues, is not the depolarization of the membrane. Do you know what the signal is for exocytosis? It's calcium, okay? Now, obviously, in a normally functioning nerve, the calcium gets into the synapse because of the depolarization. And for that, we have voltage-dependent calcium channels, okay, or voltage-gated calcium channels. So they work very similarly to sodium channels, and actually they are quite related structurally as well, okay? So all the stuff pretty much, not the inactivation bit, but the bit about moving domains and opening and closing depending on potential works for voltage-dependent calcium channels exactly alike, okay? So on depolarization of the membrane, calcium flows into the synapse and starts the process of exocytosis. Okay? Make sense? Okay, so calcium is the signal. Now, if you can put in calcium by some other means than membrane depolarization, exocytosis will go, it doesn't care about the membrane potential. The change in membrane potential is there only to allow calcium to go in. Okay? Right. So what happens, we have a neurotransmitter vesicle relatively close to the membrane. What happens in exocytosis? Now, I can see I don't have all the colored, I do actually. There are proteins both in the vesicle, in the membrane of the vesicle, and in the cellular membrane called snare proteins. Okay, it's an acronym, it's an abbreviation from some bits. Doesn't really matter what it means. Okay, there's snare proteins both in the vesicle, in the membrane of the vesicle, and in the cell membrane. I'm just going to draw some of them so that we know what we're talking about. This is very exciting. It's the first time I'm using a color chalk. Now, these snare proteins are quite logically divided into two groups. There are V snare and T snare. Can anyone guess what the letters stand for? Okay, vesicles. So those are snares on the vesicle. And T? It's target, so it's for the target membrane where the vesicles, yeah, that's a bit more difficult to guess, okay? So V snares are on the vesicle and T snares are on the target membrane. The reason why it's not called C snare for cytoplasmic or something like that, because you can have different target membranes. For example, the movement of vesicles in Golgi apparatus or ER actually goes along similar, using similar mechanisms, okay? So there's a target snares, target membrane snares, and the vesicle snares. Okay? Now, I've drawn here kind of schematically the structures. So they are alpha helical proteins which have corresponding structures in such a way that when they get close to each other, they snap together, okay, like Velcro. Okay? So once the vesicle, by some other mechanisms I'm not going to talk about, like transport on cytoskeleton, etc., etc., once they get closer to the membrane, these proteins just snap together and get the vesicle even closer to the membrane and just hold it there. Okay? So that's the first step of, I mean, it's not the first step of excitosis. Lots of things have to happen before then. But that's the first step in the bit of excitosis that we're interested in. The next step is even more interesting. This protein, which we haven't used for the attachment of the vesicle, is called, I'm just going to tell you what it's called. It's called synaptotagmine. Synaptotagmine is a protein that binds calcium. It's a calcium binding protein. Okay? So that's the connection between getting calcium in and vesicles out. Now, synaptotagmine, when it binds calcium, it increases, I mean, its affinity increases for membrane phospholipids. Okay? So it's a protein that sits there, and when it binds calcium, it really wants to bind some phospholipids. Now, what are the closest phospholipids around? Well, it's the cell membrane. So what it does, it actually creates this kink in the cell membrane. And this brings these two membranes so close to each other that they fuse together. And once they fuse, the neurotransmitter is released. Okay? Does this mechanism make sense? So first of all, we have some snare proteins that basically attach the neurotransmitter vesicle very close to the plasma membrane. And then when synaptotagmine binds calcium, so when calcium levels increase, it tugs in the cell membrane and brings the two membranes so close to each other that they fuse and release the neurotransmitter into the synapse.
So the snare proteins work without calcium, and they always try to pull it pretty close to the membrane?
Yes, it's a bit more complicated than that because I'm drawing just 4 proteins out of about 50. So there is some interaction with calcium there as well. Okay, but let's make it simple, and yes, that's how it works. Okay, I'll just mention that there is because obviously this process requires some energy. And the energy is provided by means of, and you don't have to remember that, by means of an ATPase called NSF. And that ATPase actually dissolves this complex. So once the complex is formed, the vesicle is released, we need to add some energy to untangle the Velcro so that we can do the same thing again. Okay? That requires ATP, and there's an enzyme called NSF that actually does that. Now, I'll give you the names of all the proteins that I mentioned. So we already talked about synaptotagmine. That's the calcium sensor. Okay? Now, this protein that is not really attached, that is not transmembrane, is called SNAP25. Weird names, but that's how it is. It's actually attached to the membrane via fatty acid chains, which is quite interesting. This yellow protein is called syntaxin. And which one's missing? Oh, that's called synaptobrevin. Okay, so we have SNAP25, syntaxin, synaptobrevin. Those are the Velcro bits. And we have synaptotagmine, which is the calcium sensor.
They're called as well.
Yes, they're snare proteins. They all are snare proteins. Okay? Now, the reason why I talk about these proteins, apart from trying to describe the general mechanism, is that these proteins are actually important in medicine. Because there are bacterial toxins that destroy these proteins. Okay? And the two toxins that I want to talk about are the tetanus toxin and the botulinum toxin. Okay, I will assume that everybody's heard of tetanus. Okay, so it's caused by bacterium, Clostridium tetani, which produces this toxin, and this toxin is what causes the disease. Do you know what the symptoms of tetanus are? Yeah, it's actually spasms. It's not paralysis. It's actually spasms. So yeah, you basically die. You suffocate in the end, but that's not because you can't move. It's because your breathing muscles are in a spasm. Okay. So that's tetanus. Botulinum toxin you've probably heard about, I imagine. Okay. If not in connection with poisoning or disease, probably in connection with cosmetic surgery and Botox. Okay, that's botulinum toxin. Interestingly, both of these are produced by Clostridium bacteria. So it's Clostridium tetani and Clostridium botulinum. So relatively similar bacteria. What's interesting about these two toxins is that unlike tetrodotoxin that we talked about before, the fugu poison, tetrodotoxin is a small molecule that binds to the channel and blocks it. Okay, simple. These two toxins are actually enzymes. They are pretty big protein molecules that use quite interesting trickery to get into the nerve cell. So they actually trick the nerve cell to trigger endocytosis, so that it endocytoses, because obviously they can't cross the membrane. So they trick the nerve cell to endocytose them. Once they get into the cell, they start their enzymatic activity, which is a protease. They are proteases. And what they do is they cut these proteins. Okay? And specifically, the tetanus toxin, called synaptobrevin, cuts synaptobrevin, and botulinum toxin cuts SNAP25. Okay? So a very interesting type of toxins, and actually with bacterial toxins, that's the usual way it works. They're usually enzymes that do something in the cell. These two toxins are proteases, and they cut the snares that are needed for neurotransmitter exocytosis.
Why do they have such different effects?
Yeah, very good question. Why do they have such a different effect? Botulinum toxin, which I didn't mention, but you probably know, causes paralysis. So tetanus causes spasms, and botulinum toxin causes paralysis. Well, how does it work if they pretty much do the same thing? Well, that's because the tetanus toxin has much higher affinity for inhibitory synapses and inhibitory neurons, while botulinum toxin is actually relatively specific for acetylcholine, for excitatory and neuromuscular junction, etc. Okay, so botulinum toxin will cut SNAP25 mostly in acetylcholine containing synapses and will cause paralysis, while tetanus toxin will mostly target inhibitory synapses and will cause spasms by pretty much the same mechanism. Okay. Do you have any questions? Okay, if not, that's all.